Good afternoon, students, and welcome to practice problem session week 2. It's 3 p.m. and we are just about to start. Let's wait for some more participants to join. So we can start at 3:05. So let's wait 5 minutes. Meanwhile, I'll keep admitting participants who are requesting to join this call. So if you guys are have attended the first session, you might know there are certain rules we follow in this session. Most important rule is that you have to turn off your mic and video chat uh, for all the times in the session. This will be an interactive session, but and you can communicate with me through the chat box. So the way we will progress through this session is we will solve some example questions referring to the week session of the lectures. I hope you have went through the lectures. If not, it's fine. We'll revise some concepts from these lecture series. It will have a similar pattern of questions as you are going to face in your assignment and in the exams. So we'll try to solve some example questions from the same set of lectures, and it will be a collective effort. I'll pose the questions, students will try to answer. You just have to write your answer, whatever you think is the correct answer, in the comment box, and later on we'll try to understand the rationale behind why the correct answer is the correct answer. Cool. Let's wait for three, 4 more minutes for some more students to join. We'll start at 3:05. Meanwhile, for the new joinees, please turn off your mic and video chat. You can communicate with me through the chat box. If anything urgent comes up, you can always raise your hand and talk to me. And this set of instruction is just to conduct this session smoothly, because since there will be many participants, so speaking through the mic will be chaotic. You can communicate with me through the chat box, and we'll actively communicate during this whole session. So as we can see, it's already past 3:05. So we'll begin this session without any further delay, and it will be a 2 hours long session as you might have been instructed. We'll solve a set of 10 questions in this 2 hour long session and understand the concept behind the answers. That's how the session is going to proceed. Let me just share my slides. So anything you want to talk to me or you want to communicate your answers or the about the lectures and the understanding, you can communicate with the chat box. I'll share my screen and we can start. And I hope this session will be quite interactive with you guys. I expect answers from you guys. It's okay if you do not know the answers. You can just guess the correct answer and then we'll understand the rationale behind that. So all the new students who have just joined, please turn off your video and mic, and any communication with me will be via the chat box. So you guys, I hope the instructions are clear. I have mentioned it in the messages. Interaction will be via the chat box. So I'll request every student to please turn off their video and mic. And let me just start sharing my screen. So students, just a routine check. Are my slides visible? And can you see them changing? And is my voice clear and audible? Just give me a yes or no in the comment box. Cool, cool. Yes, great. So I'll keep admitting students meanwhile they are requesting to join this meeting. So let's start with practice assignment week 2 as I have mentioned earlier. So these assignment questions, these are a practice assignment. This is not the real assignment that you'll be facing in the NPTEL course. These questions will be similarly derived from the week 2 lectures, and I welcome you all to this problem solving session. The course instructor is Professor Subramaniam from IIT Madras, and my name is Ashutosh, and I'm a PhD scholar at IIT Kanpur. So let's start with the first question. The first question on your screen: which of the following is correctly matched? So guys, here is the first question. Which of the following is correctly matched? Option A, Boveri somatic nuclear transfer. Option B, Nettie Stevens and Edmund Wilson, sex determination by chromosomes. Option C, T.H. Morgan, genomic equivalence. Option D, John Gurdon, chromosomal theory of inheritance. Here are the four options, and the question is right above you. Now you have to tell me what is the correct answer. I'll wait for a minute to collect some answers. We have almost 10 participants, and I encourage all the students to participate. You can mention your answer in the comment box, whatever you think is the correct answer. Option A, B, C, or D. This is a similar kind of pattern to your assignments and also the final exam that we'll be facing. Actually, students, you do not have to raise your hand. It's fine. You can just type your answer in the chat box and I'll be always reading the chat box. Whatever you think is the correct answer. Option A, B, C, or D, or 1, 2, 3, 4. Nisar, you can just type your answer. It's fine. Let's wait for a minute and students, please understand this question and let's move along. What do you think is the correct match out of these four options. Boveri to somatic nuclear transfer, Nettie Stevens and Edmund Wilson, T.H. Morgan to genomic equivalence, John Gurdon to chromosomal theory of inheritance. Great, I see a few answers. Ajay is answering option 2, Nettie Stevens and sex determination. Nisar is answering option 1, Boveri. Kushi and Anil are answering option B. Now let us find out what is the correct answer for this question. The correct answer for this question is option B, as many students have answered correctly. Nettie Stevens and Edmund Mincer, sex determination by chromosomes. This is the correct match. This is the correct answer to this question. Now let us see what are all these theories and scientists related to what is the actual correct option. I hope you can see the slides changing. So Boveri is not known for somatic nucleus transfer, but Theodore Boveri is a very big name in biology, and he is known for chromosomal theory of inheritance. He gave down the chromosomal theory of inheritance. And what does this chromosomal theory of inheritance say? It set up some basic rules of chromosomes, that chromosome occurs in pairs. Homologous chromosomes separate during meiosis. So homologous chromosomes which are identical, they separate during meiosis. Meiotic daughters have one of each homologous chromosome. So homologous chromosomes are those which will align nearby during meiosis, and they will segregate, and each daughter cell arising from the parent will have one of the each homologous chromosome. While fertilization restores the pair of chromosomes. So the gamete, now the meiotic daughters or the gametes here, as you can see in this diagram, the gametes here will have one of the each homologous chromosome. As you can see here, pink chromosome Y and the blue chromosome R. So each gamete has one copy of Y and one copy of R derived from the homologous chromosome. So here Y, both these pinks are homologous chromosomes, and both these blue chromosomes are homologous. So during meiosis, they will separate singly. So one copy of Y and one copy of R will go into one gamete, and the other homologous pair will segregate independently, and fertilization will restore it again as a pair of chromosomes. And genes are located on the chromosome. So these are some of the important principles that Theodore Boveri chromosomal theory of inheritance tells us. While on the other hand, on one of the options was T.H. Morgan. T.H. Morgan, Thomas Hunt Morgan, is also a big name in biology, who is known for his chromosomal linkage theory. So it's very simple theory which says that genes are linked in series on chromosomes and are responsible for identifiable hereditary traits. It just says that genes occur very closely and they are tightly linked on a chromosome, then those genes will travel together into the as a same chromosome, and there cannot be recombination. This is also one of the main theories for the chromosomal linkage theory. So genes are present on the chromosome and they are linked, and they are responsible for the hereditary traits. So the traits that is passed down from generation to generation are responsible because of genes, and they are present. They are linked in a series on chromosomes. So we can have multiple genes on a single arm of chromosome, and if they are close enough together. So Thomas Hunt Morgan gave the principle of linkage and inheritance. So if two genes are tightly linked or they are very close together in the chromosome, the frequency of homologous recombination is low. That means in meiosis, those two genes will not separate via homologous recombination. This is one of the major applications of chromosomal linkage theory. On the other hand, the third option that we saw, John Gurdon is popularly known for somatic cell nuclear transfer. John Gurdon is known for this particular experiment in which to an unfertilized egg he supplied UV light irradiation which destroyed the nucleus of the unfertilized egg. Now they only have the cytoplasm in the unfertilized egg. While on the other hand, he derived the nucleus from an adult differentiated cell. So skin cells are differentiated cell, while unfertilized egg is a unfertilized egg is enucleated because the UV light has destroyed the nucleus. Now he isolated these nucleuses, these nuclei from skin cells, and then he injected into the UV destroyed egg. To his surprise, this nucleus from a differentiated cell could still guide the formation of zygote or normal blastula, which can eventually develop into a tadpole. So for this experiment, John Gurdon was awarded the Nobel Prize for somatic cell nuclear transfer. He is famous for this experiment. The process leading to cell differentiation must involve a redistribution and interaction of constituents already present in the fertilized egg. This shows that the genetic material in the skin cells or one of the differentiated cell types is still capable of producing all the contents necessary for an organism. That means the skin cells that arose from the zygote eventually have the same genetic constitution, and everything is already present in the egg, which can lead, everything is already present in the egg, and the nucleus contains the genes which will direct the formation of organism from the egg. So John Gurdon is specifically known for somatic cell nuclear transfer. That brings us to the second option, Nettie Stevens and Edmund Wilson, who are known for sex determination by chromosomes. They, the points of their theory are: sex is determined by a pair of sex chromosomes which are different from autosomes. So we have some set of autosomes which form the somatic cells, and a different pair of sex chromosomes which determine the sex of the individual, whether it's a male or a female. They also hypothesize that X chromosome means femaleness and Y chromosome means maleness. So the correct match and the correct option is Nettie Stevens and Edmund Wilson to sex determination by chromosomes. All other were the wrong matches. John Gurdon is matched with somatic cell nuclear transfer, while Boveri with chromosomal theory of inheritance. T.H. Morgan is known for chromosomal linkage theory. That's why we have the option 2 as the correct answer. I hope it is clear to the students, the explanation part. If yes or no, just let me in the chat box. Any part you didn't understand, or is it clear? Great. Good. Now let's move on to the second question. In a similar manner, you guys have to, I'll read out the question for you. You guys can go through the options and you just let me know what you think is the correct answer. It's okay if your answer may be correct or may not. Do not need to shy out. Be confident and you can just type an answer in the chat box, right? Question 2 says there are two statements. It's a true false question. Which of the following is correct? Weizmann's germplasm theory does not support Lamarck's view of evolution. This is the first statement. Second statement, Weizmann's germplasm theory states that information is passed on to the next generation from the germ cells only. Now, which of these do you think is the correct statement? Option A, statements 1 and 2 are true, and statement 2 is the correct explanation of statement 1. Option B, statement 1 and 2 are true, but statement 2 is not the correct explanation of statement 1. Option C says statement 1 is true, but statement 2 is false. Option D, statement 1 is false and the statement 2 is true. And the last option, both these statements are false. Now you have to carefully read both these statements and match with the options. Let's wait for a minute to collect some answers. I think we are 4 or 5 students now. Some people have sadly left. But no worries, they'll join again if they wish to. You can just mention if you have already found out the answer in the chat box. Ajay has answered option 1. Statements 1 and 2 are true, and statement 2 is the correct explanation of statement 1. Okay. Nisar and Khushi are answering option 1. Good. Now let us see what is the correct answer to this question. And you students are absolutely correct. The option 1 is the correct answer. Congratulations. So statements 1 and 2 are both true, and statement 2 is the correct explanation of statement 1. Now let us see what is this Weismann's germplasm theory and how does it not support Lamarck's view of evolution, right? For the statement 1. So Weismann's germplasm theory is a simple theory which he gave. The heritable information is transmitted only by the germ cells in the gonads and not by the somatic cells. So whatever information is going to passed on to the next generation is always transmitted only by the germ cells or the germ line, the lining which forms the ovaries and the testes and which is responsible for meiotic divisions and formation of gametes, not by the somatic cells which constitute the rest of the body. So here is a diagrammatic representation of Weismann's germplasm theory. So in the first generation, we have all normal cells. So in the zygote, there is a zygote and we have separate germ cells. Some cells in the zygote are already destined for the germ cells. Now when there is a, so some cells in the zygote now form somatic cells. So this is a normal organism. Now if there is a mutation in one of the germs, if there is a mutation in one of the somatic cells, these red circles represent mutated somatic cells. So if there is a mutation in somatic cells, it can only affect the present organism. So whichever organism has the mutation, only that organism will be affected by the mutation. It will not pass on to the third generation. So the second generation came from the germ cells. Now since these are somatic cells, any mutation in that, mutations in somatic cells do not affect the germ line. This will not affect the germ line and this cannot propagate into the third generation. But whereas if you see, if there is a mutation in germline, the red bubble, if there is germline cell which has a mutation, that can now form the basis of another new zygote in a new organism. Now this zygote will have all of its cells derived from the germ cells. That's why this zygote will harbor the mutation. And since the zygote harbors the mutation now, all the somatic cells and the germ cells will be mutated. So everything is now derived from the germ cell. That's why mutation in germ cell affects the germ line. So mutation in somatic cell cannot affect the germ line, while mutation in germ cells affect the germ line. So this is a representation of how heritable information which is heritable can only be transmitted by the germ cells and not by the somatic cells. So the red somatic cells will not be transferred to the next generation. So this is in short Weismann's germplasm theory. Now what about Lamarckian theory? And how does Weismann germplasm theory violate Lamarckian theory? So Lamarck is a scientist who proposed theory based on his observations. An organism can pass on to its offspring physical characteristics that the parent organism acquired through the use or disuse during its lifetime. So this is what Lamarck said. A contrary theory to the Weismann germplasm theory, because here Lamarck is saying whatever the organism, the parent organism, is acquiring through the use or disuse of certain organs or certain pathways in his lifetime can pass on to the next generation. A beautiful example of Lamarckian theory would be a blacksmith gets strong muscle from his work. A blacksmith who works with iron and tools gets strong muscles from his work, and his sons inherit those strong muscles. This is what kind of theory the Lamarckism mean. So if the father is a strong person, then the son will also inherit strong muscle. And how does the father acquire strong muscles? Due to his blacksmith work. So Lamarckian theory states that the parent acquired characteristics via the somatic cells can pass on to the germline and to the next generation. So germplasm theory says that any changes in somatic cells cannot pass on the same to the next generation. So this is where germplasm theory does not support Lamarckian theory. Lamarckian theory says acquired characteristics via the somatic cells or whatever the body experiences will be passed on to the next generation, while germplasm theory says any changes in somatic cells will not affect the germplasm. Only mutations in the germplasm can be transferred to the next generation. That's why Weismann's germplasm theory does not support Lamarckism. So these are different theories, correct? So that's why one is focused on the somatic cell and the other states that only the germline cells are able to transmit it to the next generation. So that explains our answer to the second question. Both the statements 1 and 2 are correct, and statement 2 is the correct explanation for statement 1. Since the information is passed via the germ cells only, therefore Lamarck's view of evolution is not supported. Ajay has a question: variation, selection, inheritance, and time. What about these other points in Lamarck theory? So Lamarck theory says that variation is acquired, like vestigial organ. The best example would be the vestigial organs, or the neck in the giraffe is a very good example of Lamarckism. So the more you use it, the more it elongates. But that's not the case. So variation can be affected by natural selection, which is Darwinian theory, which is in contrast to the Lamarckian theory. So environmental factors, or any factor which is enhancing the survival of the organism, will be transferred to the next generation. So such variations will be included, but not like if you use it more, then it's going to be inherited in the next generation. So inheritance happens through the germ cells. It's it's true. Inheritance will happen only through the germ cells, but the variations that the germ cells, so the variations acquired by the individual, if any such uh, mutation is incorporated into the germ line, then that can be transferred to the next generation. I hope it's clear. Then we can move to the next question. Is it clear? You can just let me know in the chat box. So any variation in the germ plasm or any variation in the germ line cells will be transferred. So that can be a result of many things. That can be a result of any positive mutation or any environmental factors that is conferring an advantage to the individual. Let's say a mutation is improving the survivability of an individual in a certain environment. Then uh, then during course of time, during evolution, that kind of mutation is preferred and it gets incorporated into the germline, and then it can be passed on to the next generation. Yes. Yes. Yeah. Yes. Yeah. Yeah. So that variation will occur in a long period of time over. So if a person certain has certain advantages or whatever mutation, so that mutation has to go through the germline. So that is how. Yeah, 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 yes. That's why Darwin's theory is considered superior to the Lamarck's. Yeah, well and good. Let's move on to the next question. The next question is a three option fill in the blanks. In the case of cloned animals such as Dolly the sheep, they are genetically identical to the dash donor and not the dash donor. This indicates that development is driven by the nucleus. However, in some cases, cloned animals can be non-identical due to dash, for example. So options A, somatic nucleus oocyte genomic equivalence. Option B, oocyte somatic nucleus dosage compensation. Option C, oocyte somatic nucleus genomic equivalence. And option D, somatic nucleus oocyte and dosage compensation. So this is a question related to Dolly the sheep, if you guys could recall. And let's take some time to collect some answers. You can just mention the answer in the chat box, whatever you think is the correct answer. In case of cloned animals such as Dolly the sheep, they are genetically identical to dash donor and not the dash donor. That indicates development is driven by the nucleus. However, in some cases, cloned animals may not be identical due to dashed reason. Oh, great! I see already see many answers here. Kushi, Ajay, and Nisar are answering option 4. So, cloned animals are genetically identical to the somatic nucleus donor and not the oocyte donor. This indicates that development is driven by the nucleus. However, in some cases, cloned animals can be non-identical due to dosage compensation. So, this, you guys are all correct. This is the correct answer. Option 4 is the correct answer. Very good. You guys are answering very nicely. Now, let us understand the history about Dolly the sheep. So, the story of Dolly the sheep, the first uh, is the first animal to be made by this method. Cloned, Ian Wilmot cloned the Dolly the sheep. Dolly was the first mammal to be cloned from an adult cell. So Dolly was cloned from an adult cell without fertilization. So is a similar kind of somatic cell nuclear transfer. Dolly's birth proved that differentiated cells could be used to create an exact copy of the animal they came from. So what Wilmot did, took a fin dorset. This is a type of sheep or lamb, Scottish blackface. These are two different species of lambs. So mammary gland cells, the udders of the sheep, isolated cells from them, mammary cells, and these cells will act as a nuclear donor. Select a single cell. Now remove the nucleus from the donor. Collect the egg cell from the Scottish blackface egg donor. Remove the nucleus via pipetting. Now the egg cell only contains the cytoplasm, right? And then we transfer the nucleus. We combine the single selected cells using an electric shock, then formed a blastocyst. The combined cells begin to divide normally to become a blastocyst, and now the blastocyst is implanted into a surrogate mother where the blastocyst can receive nutrition from the uterus, and that blastocyst grew up into the dolly the sheep. The blastocyst developed into a fetus, and in about 5 months, a lamb is born, Dolly. That's how Dolly was born without fertilization. So we have the nucleus from mammary cell and the cytoplasm of the egg donor. So this is how Dolly was made. So that answers our first two options. Dolly the sheep. So now the Dolly the sheep is identical to the fin Dorset nuclear donor, but not to the Scottish blackface. So Dolly the sheep is identical to the nucleus from which the organism was derived. Mammary cell of fin Dorset lamb was used. So now Dolly is also a fin Dorset lamb, even if it has the cytoplasm of the Scottish blackface lamb. So Dolly the sheep, they are genetically identical to the somatic nucleus donor and not the oocyte donor. This indicates that development is driven by the nucleus. Now, what is dosage compensation? Dosage compensation is the process by which organisms equalize the expression of genes between members of different biological sexes. And the very common example of dosage compensation is X chromosome compensation. So in humans, we have females have two X chromosomes and males have one X and Y chromosome. Now, if there was no dosage compensation, then females would have higher gene product of whatever genes are present in X chromosome. That would create a biological imbalance. So that's why in mammals and in humans, of course, we have a phenomenon of X chromosome inactivation in which randomly one of the copies of X chromosome in the female is silenced and only gene product of one copy of X chromosome is active, which balances the gene product of the male and the female in the humans. And different organisms have different mechanism. In Drosophila, there is no single X chromosome inactivation in the female. Rather, the male X chromosome, is hypertranscribed. Now, the male chromosome is transcribing two times the genes. So, double amount of gene product of the X chromosome is now compensating the inactivity, the absence of another X chromosome. So two chromosomes here in female and here one X chromosome in the males produce doubly the amount of genes. And in C. elegans we have hypotranscription of both X chromosomes. So it's like these X chromosomes became half and half to match the single X chromosome in the males. So if such dosage compensation happens, if happened during Dolly the sheep cloning or any kind of cloning, then it won't be identical to its parent or to the identical to its donor, nucleus donor, because the donor might have random, let's say the dolly, the sheep is a mammal which undergoes X chromosome inactivation. The donor might have a random X inactivation, and now dolly, the sheep, or the, the fetus born out of somatic cell nuclear transfer can have random inactivation of the other X chromosome, which might change the genetical identity, which might change the identity between the donor and the fetus. So that's why dosage compensation. In some cases, cloned animals can be non-identical due to dosage compensation, X chromosome inactivation. Is this clear to the students? You can just let me know with a yes or no. You guys have answered very correctly. So in the sex chromosomes, yeah, we have unequal number of chromosomes. So whatever genes, Yeah, because yeah, whatever. Yeah, yeah. But the blood group system that you were talking about is present in the somatic, the autosomes. So in a same individual or in the same species, the number of autosomes are same. So we have 22 pairs of autosome. The female will have and the male will have same. But the only difference that we have is in the sex chromosome. So if a blood group gene is present in the sex chromosome, then it might be doubly, the gene product will be doubly produced, which is absent in the male. That's why, or whatever gene that is in the X chromosome needs to be deactivated. One X chromosome is deactivated so that it can compensate the, so male and female will have equal amount of gene product. So blood grouping, the one I think blood grouping you were saying about allelic conformation, because A and A will produce the A blood group. So whatever genes that are present in the somatic or in the autosomal chromosomes, the other 22 pairs of chromosome will be similar. Even if we have a single pair of chromosome or double pair of chromosome during, so we have 22 pairs of chromosome, whatever gene in the autosomal chromosome will be similar. The only difference among a biological sex in a certain species is the male and female is the X chromosome. Females will have two X chromosome and males will have one X chromosome, or vice versa. Many organisms have different chromosomal sex determination. Since they have an extra copy of chromosome, uh, now the genetic dosage compensation will compensate the gene products. That's how we have one chromosome inactivation, or each of the male chromosome, let's say hyper transcription or C. elegans, we have half and half production. So any gene, if blood group gene is also present in the X chromosome, then it can be inactivated. So that's why let me give you another example to make it clear. The X-linked disorders that we have, there are many genetic disorders which are X-linked. So a mutation in one of the copy of gene is sufficient. It's X-linked dominant disorder. So sometimes females are just carriers of this disease as for recessive disorders, but sometimes even dominant disorders, the female is not affected because the chromosome, the X chromosome harboring the mutant copy of the X-linked dominant disease is now silenced. So only the normal healthy gene in the functional chromosome is active. That's why even the female having a mutant gene for a disease in the inactivated X chromosome will not harbor the disease. Great. I hope it's clear. Now let's move on. Yes, yes, yes. Yeah, yeah. But color blindness is a X-linked recessive disorder. Yeah, yeah. No. Yeah, yeah, yeah. It is random. It is random. Yes. Yeah, but the female harboring the mutation, The female harboring the mutation won't. The next generation of females can suffer from color blindness, because now the germline which has these mutant X chromosome and which is not inactivated, which is now active with the mutant, then that can go and form in the next generation. The progeny of the female can form the color blindness. So it can be, if the progeny. I think you are not clear. Okay, let me get back to you after in the next class, or in the solving questions of whatever question you want to ask about dosage in compensation. I hope I answered, but maybe you want demand more answer. So let's move on to the next question, and I'll try to just you just have to remind me in the next class. I'll read it up and try to come and explain. Okay. The fourth question: which of the following is not present in a eukaryotic gene? This is this is a question about eukaryotic gene construct. Which of the following is not present in a eukaryotic gene? Option A, 3' UTR. Option B, promoter. Option C, poly A tail. Option D, introns. So let us collect some answers now. Now, which of these is not present in a eukaryotic gene? I think people can answer this. It's an easy question. You just have to know the simple definition of a gene. Then you will be able to figure out which of these is not present in a gene. Ajay and Khushi are answering option 3, poly A tail. And poly A tail is the correct answer. Poly A tail is not present in the eukaryotic gene, while the other three elements, 3' UTR, promoter, and introns, are part of the eukaryotic gene. So here is a representation of how a gene looks like. So eukaryotic gene, the DNA segment which can instruct the formation of proteins in eukaryote. That is the eukaryotic gene. It's a DNA segment, and DNA segment will have these unique DNA segment will have different regions. Upstream enhancers at the 5' end. So we have 5' end and the 3' end to the DNA. We have promoter regions, data box regions, 5' UTR, the exons, introns, and the 3' UTR. So this makes up the whole gene in a eukaryotic gene as per se. The promoter is a region where transcription factors will bind. Data box is a region where polymerase binds. 5' UTR is a region between the data box and the first exon, and exons are the coding sequences, followed by intron which are non-coding, and 3' UTR. So 3' UTR, the option 3' UTR, promoter, and introns are all part of coding region or the DNA eukaryotic gene. But poly A tail is a post-transcriptional modification which occurs only in the mRNA. Now, when the transcription process is complete, polymerase has transcribed the RNA. The RNA will be capped by a 5' cap, 7-methyl guanosine cap, and a poly A tail will be attached at the 3' end. So this is not a DNA structure. This is an mRNA structure. That's why it is absent in the eukaryotic gene. So another simplified example of RNA processing in eukaryotes. So once we have the primary RNA transcript, We have 5' UTR, exons, introns, and 3' UTR, which are then processed. The introns are removed, exons joined together. The 5' end has a guanosine 7-methyl guanosine cap and a poly-A tail added. So poly-A tail is a post-transcriptional modification which is only present in the mRNA and not the DNA. And the DNA is what constitutes the eukaryotic gene or any gene. I hope this was simple and clear. So poly-A tail is a mRNA modification, and promoter, UTR, and introns are present in the DNA as well. You can just tell me if it's clear, yes or no, and we can move to the next question. No screen. Oh, could you not see my screen? Is it not visible? Ajay, but Kushi says yes. Ajay, is it the screen visible to you? You can just type yes or no. Yeah, yeah, fine. Now the fifth question, identify A and B. A can be used to identify whether binding takes place between nucleic acids and proteins. So, and B can be used to identify which sequence is larger fragment of DNA is binding to a protein. Which sequence in a larger fragment of DNA is binding to a protein? And option A, gel mobility assay for A. B, immunostaining. Option B, A, DNase protection assay. B is gel mobility assay. Option C, A is gel mobility assay, B is DNA protection assay. Or option D, A is immunostaining and B is RNase protection assay. So you have to identify the procedures. A can be used to identify whether binding takes place between nucleic acid and proteins, and B can be used to identify which larger sequence fragment of DNA the protein is binding to. Kushi has answered C. I think Ajay is not able to see the screen. What did just give me a minute. I'll share my screen again. I'll escape and share my screen. Can you see the slides changing? Oh, I have just displayed the answer for the question, and Kushi has answered correctly. So option C is the correct answer. A is gel mobility assay and B is DNase protection assay or DNase fingerprinting. So let's learn a bit about these two processes. Gel mobility assay is a technique used to study DNA or protein RNA interactions. So if a protein is binding to a DNA or RNA or any nucleic acid, this is a technique to identify that. This procedure can determine if a protein or a mixture of proteins is capable of binding to a DNA or RNA sequence. And this relies on the principle that the more the load on the DNA, the slower the DNA becomes. So if you have free DNA, here is a gel schematic diagram. If you have free DNA, the DNA will be fast enough running through the gel, and we can locate the unbound DNA here, unbound. Now if a DNA is bound by a protein, so if you want to test whether a protein can bind a nucleic acid or an RNA, we take that sample protein, we add it with the DNA. Now if DNA is bound by the protein, the DNA will be slower because now it has more load. Proteins are sitting on the DNA. It will move slowly through the gel and the band will appear somewhere here. And this is the shift which is measured, the unbound DNA and the protein bound DNA. And another assay the gel mobility assay can be improved by an antibody that recognizes the protein. If it is added to this mixture, it will create an even larger complex with a greater shift. This method is referred to as super shift assay and is used to unambiguously identify a protein present in protein nucleic acid complex. So now if we know what protein we are going to test and we have an antibody against the protein, we can apply the antibody, DNA, protein and antibody. Now this complex is bigger, the DNA is slower and the shift will be higher. Shift from the unbound DNA and the band where the antibody bound with the protein and the DNA. If ever the protein is interacting with the DNA, it will be able to shift the DNA, this band shift, which is then observed as the uh, ability of protein to bind and recognize nucleic acid. So similarly here we'll have a single band here at unbound DNA. Protein bound with DNA will occur somewhere in the middle, and antibody plus protein plus DNA will occur at the topmost band because it's heavier, more complex interactions, and the DNA is slower. So this is gel mobility assay, can be used to identify whether binding takes place between nucleic acid and protein. If ever protein is binding to the DNA, it will slow down the nucleic acid or RNA. Now in the next option, DNA's protection assay was the correct answer. So what happens in this assay? This assay detects DNA protein interaction using the fact that a protein bound to DNA will often protect the DNA from enzymatic cleavage. So if a long piece of DNA, large fragment of DNA is wrapped around a protein, or a protein has the ability or a DNA binding domain, region or the region where the protein is interacting with the DNA will be protected from enzymatic cleavage. So this technique takes advantage of this fact. This makes it possible to locate a protein binding site on a particular DNA molecule. So the simple procedure, we have genomic DNA, labeled template DNA containing protein binding site. We amplify certain regions of DNA, whichever region we want to check whether there is a protein which can bind to the DNA. We add our proteins of interest and we cleave the DNA. So we use DNase deoxyribonuclease which will cut the DNA. Now if you see, if a bulky protein is binding the DNA, then the DNA which is bound to the protein cannot be cut, is not accessible to the deoxyribonucleases. So where we'll have fragments where the DNase would not work. This method uses an enzyme deoxyribonuclease to cut the radioactively end labeled DNA, followed by gel electrophoresis to detect the resulting cleavage pattern. So here if you run this treated protein DNA complex in a gel, we'll see the protected fruit wind. In a DNA when there is no protein, we'll have n number of fragments depending on the enzyme. So deoxyribonuclease depending on the sites in which it cuts the enzyme. So there's any restriction endonucleases, whatever specific site. How we know that there are 10 sites, let's say in a DNA, so we'll have 10 different fragments. But now in the presence of protein, we miss certain fragments, which is known as the footprint of the protein. DNase protection assay. The protein is binding to those areas in the DNA which is not accessible by the enzyme. That's why we are not seeing those fragments in the gel. So this is a DNase protection assay or a DNA fingerprinting assay in which larger fragments of DNA can be recognized which are bound by some proteins using the fact that protein bound DNA is not susceptible to enzymatic cleavage. I hope this is clear to the students. You can just let me know with a yes or a no. So we learned about two different assays, gel mobility assay and DNase protection assay. Both are assays to determine nucleic acid protein binding and the sites to which the protein recognizes. Yes, great, great. Now let's move on to the next question. Question number 6. Which one of the following is factually correct? There are certain statements and you have to identify which is correct. And this is about enhancers and transcription factors. Both enhancers and transcription factors work in a modular fashion. Option number A. Option B. Enhancers work in a modular fashion while transcription factors have combinatorial functions. Yes, Ajay, you were saying something? Oh, you can, yeah, you can just mention in the chat box. I'll always be reading the chat box. So option A, both enhancer and transcription factors work in a modular fashion. Option B, enhancers work in a modular fashion while transcription factors have combinatorial functions. Option C, transcription factors work in a modular fashion while enhancers have combinatorial functions. And option D, both of them are combinatorial in function. It's a tricky question. You have to understand what is modularity and what is combinatorial property. Then we can reach to the right answers. Whether enhancers are modular or combinatorial, whether transcription factors are combinatorial or modular or both. Ajay says option 4, which was telling already. Khushi is answering option B. Now let's check what is the correct answer. Correct answer is option B. So Khushi has answered correctly. Enhancers work in a modular fashion while transcription factors have combinatorial functions. So enhancers are modular and transcription factors are combinatorial. Now let us understand what are these terminologies. Enhancers, what are enhancers and transcription factors? Enhancers are classically defined as cis-acting DNA sequence that can increase the transcription of genes. So cis-acting DNA sequence means they are present cis to the gene. So wherever the gene is present, the enhancer is present cis to that. Cis means uh, in the same orientation. It can be present on the same chromosome, can be present on a different uh, chromosome, many base pairs away. Enhancer's location can be very dynamic. They generally function independently of orientation and at various distances from the target promoter. So, enhancer can enhance the transcription of the genes. That's why it's called an enhancer, and it's a DNA element. While transcription factor is a protein. Transcription factor is a protein that controls the rate of transcription of genetic information from DNA to RNA by binding to promoters. So the function of transcription factor is to bind a promoter and recruit the RNA polymerase to the data box of the gene. So whatever the promoter uh, is, whatever gene is under the promoter, RNA polymerase will transcribe the gene in the presence of the transcription factor. So recruiting of RNA polymerase is a critical step in the rate of gene transcription. Transcription factors work alone or with other proteins in a complex by promoting as an activator or blocking as a repressor the recruitment of RNA polymerase to specific genes. So here we see the combinatorial property of transcription factors. Transcription factors can often work in complexes with activators and repressors which can change the outcome of the target gene expression. So here is a diagrammatic representation. We have enhancers, cis-acting element. So here is the gene and enhancers are cis to them. Transcription factors are the proteins which sit on their promoter region and recruit the RNA polymerase with the help of enhancers, of course, and RNA polymerase can then turn on transcription. Now, a comparison between modularity and combinatorial nature of the transcription factor. Modularity of enhancers mean that what I mean by this term, enhancers can act independently of the genes they regulate and their effects are context dependent. A single enhancer can influence expression of multiple genes, and multiple enhancers can regulate expression of a single gene. So this is the modularity concept. In a context-dependent manner, a single enhancer can influence expression of multiple genes because the cis element may be present on a single chromosome can affect all the genes or many of the genes that needs to be transcribed which have the specific promoter, right? And what does this enhancer do? They loop in the DNA and form a active complex with mediators, transcription factors, and histone modifiers that opens up the gene for transcription. So histone modifiers will open up the gene for transcription. Mediator proteins which help in recruiting of transcription factors as well as RNA polymerase. So these enhancers will bridge the mediators and transcription factors and help in recruiting the polymerase to the promoter region. This enhancer modularity is essential for integrate regulation of gene expression during various biological processes such as development, tissue-specific expression, and response to external stimuli. So the same set of genes, let's say many toolkit genes, many genes which are really important for any organization. Let's say any cell signaling pathway, very important. So the similar kind of genes can be present under different enhancers. So the modularity, the change in enhancer can change the expression of gene in a tissue specific manner. Let's say a important signaling molecule which is Wnt signaling. You guys must have heard many important or GPCR signaling. So the genes for GPCR signaling, they will have a certain specific promoter, but they can have multiple enhancers. An enhancer which is eye specific, an enhancer which is hand specific, an enhancer which is leg specific. So at specific tissues, different enhancers can activate the transcription of the same gene. So different enhancers can regulate the expression of a single gene, or a single gene can regulate the expression of multiple single enhancer can influence the expression of multiple genes. By having modular enhancers, the cell can achieve precise and intricate control over gene expression patterns without interfering with the core gene structure. So uh, enhancers are cis-acting elements, and they act epigenetically. They bring in the factors necessary for initiation of transcription. That's why the cell can achieve control over gene expression without interfering with the DNA itself. So there is no change in actual gene structure or the coding sequence of the gene. The presence and absence of an enhancer or the can be essential for regulating the gene. Now, combinatorial nature of transcription factors refers to the fact that they often work together in complex combination to regulate the expression of genes. Many genes are regulated by the coordinated binding of multiple transcription factors to their promoter or enhancer regions. So transcription factors can also bind to enhancer regions through the mediators or directly, and many different transcription factors can bind together to coordinate the expression of genes. Each transcription factor recognizes specific DNA sequence, and combination of transcription factor binding to these regulatory elements creates a unique regulatory signature for the gene. Transcription factors can interact with each other, leading to synergistic or antagonistic effects on gene expression. So, transcription factor is never alone. It is accompanied always by mediator proteins, by deacetylases, by other transcription factor, and they form a complex. If you see the eukaryotic uh, transcription factor complexes, they have many components like TF2D, TF2A, TF2B, which have different functions. They assemble together as a complex, sit on the promoters, recruit the RNA polymerase, and start the process of transcription. Thereby, we say that transcription factor have a combinatorial nature, while modularity is one-to-many relationship. Many enhancers are present for a single gene, which can influence the expression of a single gene in a tissue-specific manner, or a single enhancer can influence uh, multiple genes. The single enhancer, though because it is a cis-acting element, can influence whatever genes present on the same chromosome. It can influence the looping and bending and increase the gene expression of the multiple genes. And here is an example of modularity in enhancers, the example that I was giving earlier. Gene A expressed in brain and limb. So we have this gene A, which have two enhancers. So multiple enhancers controlling the expression of single gene, brain specific enhancer and a limb specific enhancer. So what will happen in brain? The brain specific enhancer will form a complex with mediator, and they will have brain expressed transcription factors which will bind to the brain specific enhancer. So transcription factors are very unique. Transcription factors are very unique to the tissue. Transcription factors will bind to the brain specific enhancer while not to the limb specific enhancer, and then recruit the mediator and RNA polymerase which will express the same gene A in the brain. While in the limb, transcription factors can use limb specific transcription factors can use the limb specific enhancer to recruit RNA polymerase to express the same gene in the limb. So if a gene if required in the limb and in the brain, they can have different enhancers, and different transcription factors can recognize the different enhancer and transcribe the gene expression. So this is modularity of enhancers. Ajay is requesting for the question slide again. Yeah, I think yes, I can do that. Which one of the following is factually correct? This was the question slide. Both enhancers and transcription factors work in a modular fashion. Enhancers work in a modular fashion while transcription factors have combinatorial function. So there are slight differences between modularity and combinatorial. So transcription factors are always a complex of different proteins, while enhancers they have multiple one to many and many to one relations. So that's how they are generally working. I hope it is clear to the students. If yes, just let me know. We can move on to the next question. Great. The seventh question here is before you. Which of the following is correct? It's again a true or false statement, similar to the earlier ones that we had. DNA methylation always inactivates gene transcription. Statement 1 says DNA methylation always inactivates gene transcription. And statement 2 says effect of histone tail modifications depends on the location at which the modification takes place. I hope you can see the questions. If there is any problem, then yeah. DNA methylation always inactivates gene transcription, statement 1, and the effect of histone tail modifications depends on the location at which modification takes place. Now there are various true false statements. Option A, statement 1 and 2 are true, and statement 2 is the correct explanation of statement 1. Option B, statement 1 and 2 are true, but statement 2 is not the correct explanation of statement 1. Option C, statement 1 is true, statement 2 is false. Option D, statement 1 is false and statement 2 is true. And the last option, both the statements 1 and 2 are false. You have to read the question very slowly and carefully, and only then can you reach the answer. Let's wait for some time and collect some answers. Ajay says 3. Is it the previous question or the correct question? I think it's the correct question. Statement 1 is true and statement 2 is false. Khushi is saying option D. Statement 1 is false and statement 2 is true. Okay. The other students can also answer. Do not feel shy. You can answer, just have to mention the option in the comment box, and we will try to understand the explanation here. Option 4, Ajay has changed his answer. Statement 1 is false and statement 2 is true. Now let's see which is the correct option, correct answer. Oh, no, Ajay, there are 5 options here. So Khushi is answering D, and Ajay is answering 4. Ajay is also answering D. The correct statement is D. Well done, Khushi and Ajay. Statement 1 is false and statement 2 is true. Right? Now let's understand a little bit about DNA methylation and histone tail modifications. So DNA methylation generally is repressive, but based on sites of DNA methylation in a locus, it can serve as a repressive or activation mark for gene expression. So it can work both ways based on the site of DNA methylation. In a crosstalk with histone modifiers, DNA methylation can consequently either inhibit binding of the transcription machinery or generate a landscape conducive for transcription. So methylation can work both ways in a crosstalk with histone modifiers. So histone modifiers open up or close up the chromatin, providing accessibility to the transcription factors and the RNA polymerase to whether turn on a gene transcription or turn off a gene transcription basis on the chromatin, the chromatin opening of the chromatin and closing of the chromatin, right? And DNA methylation can recruit different histone modifiers based on the context. Now, one such example where DNA methylation leads to activation in cancer cells, aberrant DNA methylation patterns can lead to activation of certain oncogenes. In cancer cells, aberrant DNA methylation, there are abnormal DNA methylation. Specifically, DNA methylation sometimes present in introns can lead to activation of certain oncogenes. And another gene, interferon gamma, recent studies have showed that interferon gamma DNA methylation at enhancer region was found to promote transcription factor binding, leading to enhanced gene expression. So here the DNA methylation is in the enhancer region, which now recruits, which can now recruit a transcription factor and lead to enhanced gene expression. And for your information, IFN gamma is a cytokine which is responsible for immune cell activation, mostly the macrophages and natural killer cells. They are these cells are major part of our immune system. Macrophages are phagocytic cells, and killer cells uh, killer cells do cytotoxic killing. So they IFN gamma stimulates natural killer cells and macrophage cells, and this IFN gamma gene, the enhancer of this gene, when methylated at certain regions, was found to promote transcription of this IFN gamma gene. So DNA methylation is not always repressive; it can be activatory, depending on the sites and locus where the methylation is occurring. Because there are many proteins which can recognize these methylation marks, and it depends on the nature of proteins whether their gene transcription will be activatory or repressive. So here, if a transcription factor is recruited to the methylation mark in the enhancer, it can be activatory, but generally DNA methylation at the CPG islands or the uh, CG repeat motifs are repressive because it recruits histone proteins and coils of the DNA, right? But it generally it's not the case. There are exceptions. Interferon gamma is one such example. And now coming on to the example of histone tail modifications. If you remember the statement 2, the effect of histone tail modification depends on the location at which the modification takes place. It's very important. The location of the residue or the location of the residue in histone tail determines the nature of activity the modification is going to carry out. Histone methylation can have different effects depending on specific lysine or arginine residue being modified and the number of methyl groups added. So there can be variability in lysine and arginine groups, the position of the amino acid or the preference of amino acids, and also to the number of methyl groups that are added to the amino acid. Methylation of histone 3 lysine 4 H3K4Me. So fourth position lysine in the histone tail of histone 3 is generally associated with active gene expression when it occurs at gene promoters. However, H3K4 methylation at gene bodies can also play a role in regulative alternative splicing and elongation. So at gene promoters, if histone 3 tail lysine 4 is methylated, it can activate gene expression. On the other hand, methylation of histone 3 tail lysine 27 trimethylation is a repressive mark and is associated with gene silencing when it occurs at gene promoters or enhancers. So you see the subtle difference. The fourth lysine, when monomethylated, is active mark, but the 27th lysine, if trimethylated in histone tail, is a repressive mark. Because the so here is a diagram, representative diagram showing H3K27 trimethylation, which activates PRC2 polycomb repressive complex proteins, which can then produce a repressive heterochromatinize the DNA, while K4 monomethylation will inhibit recruitment of polycomb repressor proteins. This will lead to active chromatin, the active state of chromatin. So here we can see different position of methylation. The number of methyl groups added can recruit or facilitate the entry of different proteins and can affect the heterochromatinization or euchromatinization. So histone methylation is very much dependent on the location at which the modification takes place. And DNA methylation, as we have already seen, may not always may not always inactivate gene transcription. There are some instances in which it activates gene transcription. So option D is the correct answer. Statement 1 is false and statement 2 is true. I hope it's clear to the students. Just let me know quickly. Yes, great, cool. Now we're coming towards the end of our problem solving session. 2 or 3 more questions, question 8, 9 and 10, and we are done. So I will encourage all the participants to answer, although we have very few participants. No need to shy out. Just try to guess the answer based on your logical understanding, and we'll get to the correct answer. So please try to participate. You just have to mention the correct option in the comment box, right? Now this is a question about enhancer traps. Which of the following is not used in enhancer traps? You have to see carefully. Which of the following is not used in an enhancer trap? Option A, Cree recombinase. Option B, a strong promoter. Option C, a reporter gene. Option D, transposable element. Which of these do you think is not used in an enhancer trap? Now for answering that, you guys have to know what is an enhancer trap and what are its components, and one of these components does not belong to the enhancer trap, right? Khushi has answered option B. Ajay is saying 2. Can we have 2 more answers, guys? What do you think, the other 2 students, Pushpa and Ashwin? What do you think is the correct answer? You can just mention, right? Which of the following is not used in enhancer traps? Okay. We'll go on with the current number of answers. Let us see what is the correct option. You guys are right. The correct option is option 2, strong promoter. A strong promoter is not used in enhancer traps, while all the other three components can be used in an enhancer trap. Now let us understand what is an enhancer trap and how does it work. An enhancer trap is an insertional construct that contains a weak promoter upstream of a reporter gene. So it's a simple element. So here is the trapping element. We have a simple promoter, weak promoter. It has to be a weak promoter and a reporter gene, which might be beta galactosidase, LAGZ expressing genes, GFP, or any other such reporter. And we have transposable element. An enhancer trap is a insertional construct that contains a weak promoter of a reporter gene and a transposable element. So the transposable element will facilitate insertion of this whole construct randomly into the genome, randomly anywhere. So what we do in such enhancer trap experiments or in the technique, the promoter is too weak to generate anything other than a minimal expression on its own. So we have a weak promoter. Weak promoter is essential because a weak promoter will only lead to minimal transcription on its own. So there will be less reporter gene activity. Now what will happen if this weak promoter construct inserts itself into or in the vicinity of a powerful enhancer? If the construct inserts in the vicinity of an endogenous enhancer which is powerful, which can increase the gene transcription, this may activate the gene and generate a strong and specific expression pattern. So what will happen if the transposable element having the promoter, minimal promoter reporter inserts into an area nearby an endogenous enhancer? Now there will be active transcription of the reporter and we can see translation of the reporters. We can see more of reporter. So if we have a GFP construct, we will see more green fluorescence if by chance it inserts into a region which has a good enhancer, which has a high profile enhancer. So a high fidelity enhancer can even drive transcription of a weak promoter. We have a weak minimal promoter here, but a high fidelity enhancer will even transcribe, even produce high gene transcripts of the reporter in presence of a weak promoter. This technique is called enhancer trap. So we need a weak promoter. A strong promoter won't do, because then we cannot identify the presence of an enhancer or whether the construct has reached the vicinity of an enhancer, right? Now how do we use a practical experiment where we use Cre recombinase, transposable elements and the reporter gene. In an enhancer trap experiment, a reporter gene like LAGZ or GFP, which I have told, is placed downstream of a minimal promoter and flanked by LOXP sites. So Cre-LOXP is a recombinase system in which LOXP is the site which are recognized by a recombinase called Cre, and then Cre can cut the LOXP sites and cause a deletion, excision or inversion depending on the orientation of LOXP sites. So it is a recombinase system. We provide a site where the recombinase can go, cut the DNA and induce recombination of the DNA. So when the enhancer trap construct is integrated near an enhancer element, enhancer's activity can drive the expression of the reporter gene. Even if it is a minimal promoter, the strong enhancer can drive the activity. And to investigate the enhancer's function further, Cre recombinase is introduced into the system. Now that we have the whole enhancer construct, the reporter construct flanked by LOXP site, Cre recombinase will remove the reporter system, remove the reporter construct when once expressed. Cre recombinase recognizes the LOXP sites flanking the reporter and catalyzes the recombination between these sites. This process can either excise the reporter entirely or reposition it, thereby altering its expression pattern. By doing so, researchers can manipulate the activity of enhancer and study its role in gene regulation and development. So here the LOXP sites which is harboring the LOXP sites, which is present in the reporter gene. Now that we have a Cree system, Cree will excise the GFP protein or whatever the reporter gene, and the signal will decrease, which will then tell us the activity, report the activity of the enhancer. We can have even LOXP sites at the site of enhancer. By removing the enhancer, we can show that the expression is going down. So these kind of systems have been used, and the basic concept of enhancer trapping is a transposable element with a reporter gene and a minimal promoter. If it is in a presence, if it is in the vicinity of a powerful enhancer, then the reporter gene activity will increase. So the correct answer, as mentioned by students, a strong promoter is not used in enhancer traps. I hope it's clear to the students, and we'll move on to the ninth question, which is the match the following question. Students have to see carefully what options will match what. On the left hand side, we have epsilon globin, gamma globin, NRSE, pioneer transcription factors, leucine zipper. On the right hand side, we have silencer, DNA binding domain, expressed only in fetus, bind to repressed chromatin, expressed only during development. Now, which of these do you think is the correct match options? I hope you can see those options in the screen. Then I do not have to read it out for you guys. Oh, Ajay is very quick in answering. He has already answered option 4. So let's wait for others to answer. Guys, we are already at the ninth question. After this, we just have one more question and we'll be done with the session. So please try to answer at least these last few questions and try to refresh your memory of the lectures and your understanding. So what do you think is the correct match for these columns? Epsilon globin and gamma globin, NRSE, pioneer transcription factors, leucine zipper. I can see some answers. Okay, Ajay is mentioning 4. Pushpanjay is saying 3. Kushi is saying option B. Okay. Now let us see a few of these options. So in these kind of questions, you have to be a little clever. You do not have to get all the options, all the match the following right. You do not have to know everything. Even if you have 3 out of 5 matching the correct, then the rest will be given in the option correctly. You can find the option, right? So let us see what is the correct option. The correct option is option 4. So Ajay has answered correctly. Epsilon globin will match to option match the following 5, expressed only during early development. Gamma globin expressed only in fetus. NRSC is a silencer. Pioneer transcription factors have DNA binding domain. Sorry, bind to repressed chromatin, and leucine zipper have a DNA binding domain. It can be quite confusing, but you just have to be sure about maximum number of Mazzi followings, and then you can tick the correct option, right? So option 4 is the correct answer. Yeah. I will come to that. I have a slide on epsilon and gamma globin. So early development, fetus is a stage. Early development means in zygotic, blastula, and those stages. And fetus is a certain stage. So during those, we have the expression of epsilon globin. So if you see, alpha globin and beta globin are the two proteins. Uh, are the two proteins are yeah which make up hemoglobin, and they have many locuses, many different kinds of alpha globin as well as beta globin. We have epsilon, we have gamma, gamma A, gamma G, we have delta and beta, and they are expressed in different time points and in different area. So embryonic hemoglobin and fetal hemoglobin. So fetal liver expresses alpha 2 gamma 2 hemoglobin. So embryonic to fetal switch happens in 6 weeks. So before 6 weeks we have the expression of epsilon type of beta globin along with zeta alpha globin. This is the constituent of hemoglobin chain. Hemoglobin is a tetrameric protein consisting of two molecules of alpha globin and two molecules of beta globin. So before 6 weeks is the embryonic stage in the yolk sac, where we have the presence of epsilon globin, and in the fetus stage is between 6 weeks to the gestation period. That is the fetus stage. In fetus stage we have alpha 2 from the alpha globin locus and gamma 2 of the beta globin locus, the fetal liver, and bone marrow. In the adult we have alpha 2 beta 2. So this is the common hemoglobin that we all know. Epsilon globin expressed as a part of embryonic hemoglobin during early stages of development. So before 6 weeks of gestation, gamma globin expressed only in the fetus. So after 6 weeks we can call the embryo a fetus. Gamma globin is expressed there in the fetal liver and epsilon globin in the yolk sac. While zeta globin and alpha 2, so alpha globin from the locus is expressed in the fetal and embryonic respectively. Now one of the other options match the following, which is easier to remember, is about leucine zipper and the DNA binding domains. Leucine zippers are DNA binding motifs or proteins which have these leucine zippers uh, are said to be DNA binding. Leucine zippers are a dimerization of the motif called BZIP or basic region leucine zipper class of eukaryotic transcription factors. The BZIP domains is 60 to 80 amino acids in length with a highly conserved DNA binding region, basic region, and a more diversified leucine zipper dimerization region. So two BZIP domains will dimerize to form a leucine zipper, and this is the structure how it looks like. And where does this leucine zipper binds? This leucine zipper binds the basic leucine zipper binds via the basic and amine residues of certain amino acids in the basic domain such as lysine and arginine. So the DNA binding domain have a lot of lysine and arginine, and they bind to the DNA. Now it is pretty cool to notice that lysine and arginines are basic or positively charged amino acids which are present in the protein, which can easily bind and interact with the negatively charged DNA backbone or the phosphate backbone in the DNA. That's why we have facilitated this interaction. Leucine zippers have positively charged amino acid recognizing the negatively charged phosphate backbone of the DNA. These basic residues interact with the major groove of the DNA forming sequence specific interaction. Sequence specific interactions, major group of DNA, and the basic principle behind it. Basic amino acids of the leucine zipper like lysine and arginine bind the uh, negatively charged phosphate backbone in the DNA. I hope it's clear to the students. Daya is asking, can we access previous sessions? Yes, Daya. So all these sessions are recorded, and you will also have access to the slides. These recorded sessions will be uploaded to YouTube, and you will also have the YouTube link sent by the NPTEL team. Or I can do one thing, after we finish this session, I can copy paste the YouTube link in the comment section. You can just go and watch the previous session. Do not worry if you have missed due to certain reasons, but you definitely should not regularly miss, because these sessions will help you in solving the actual problems when you face the assignment and the examination, and also will brush up your lecture series. So you will get these slides. I'll upload these slides in a drive, and you will get the link of the drive folder as well as the YouTube link to which the recordings of these sessions will be uploaded. Yes, I will definitely send the link in the comment box. You can copy it from there. After we finish the last question of this session, which is on the screen now, and I hope that I expect all the people to answer this question. This is about true false statement. Which of the following statements are true? Genomic imprinting involves sex specific methylation. And statement 2, X chromosome inactivation in humans is non-random. And statement 3, Prader-Willi syndrome is caused by deletion in the paternal copy of a gene, while Angelman syndrome is caused by deletion in the maternal copy. We'll take some time and look at the options. Which of the following statements are true? Is it only 1, 1 and 3 only, 1, 2 and 3, and 2 and 3 only? Ajay and Khushi have both answered option B. Can I expect two more answers from the other two students? This is the last question of the session. Please put in some effort. I know you can answer this question. Genomic imprinting involves sex-specific methylation. X chromosome inactivation in humans is non-random. And Prader-Willi syndrome is caused by deletion in the paternal copy, while Angelman syndrome is caused by deletion in the maternal copy. Okay, without wasting any time, seems like students are shy. Option B is the correct answer. Khushi and Ajay both have answered correctly. Statement 1 and 3 are true. Genomic imprinting involves sex-specific methylation, while X chromosome inactivation is random, it's not non-random, and Prader-Willi syndrome caused by deletion in the paternal copy, while the Angelman syndrome is caused by deletion in the maternal copy. Now let's have a brief introduction to genomic imprinting. Genomic imprinting is a process of silencing genes through DNA methylation. And the repressed allele is methylated while the active allele is unmethylated. So as we were talking about DNA methylations in CPG islands, so we have certain CPG islands which when methylated can lead to transcriptional silencing of certain genes. And the pattern, genomic imprinting is an epigenetic phenomena that results in differential expression of genes depending on whether they're inherited from the mother or the father. And the pattern in which the mother has its genes imprinted or certain genes silenced will be inherited in the offspring as well as the fathers. It depends on what copy, depends on the copy of chromosome that the offspring is receiving, whether it's from the silenced gene from the mother and the father. So both these signatures are imprinted into the fetus or in the offspring. One of the key mechanisms underlying genomic imprinting is DNA methylation, as we have talked. Specifically, DNA methylation marks are established during gametogenesis and are differentially maintained in resulting male and female gametes. So, male and female gametes derived from the individuals carry the individual's genomic imprinting marks. So, whichever genes are imprinted in the father will be carried forward in the sperm and in the egg as well. Egg will come from the mother, which will be distinct from what is in the father. So we have some non-imprinted genes from the male and the female giving transcription of mRNA here, and while imprinted genes, maternal imprinting, where some genes in the maternal chromosome are imprinted or are silenced. Thereby we see only expression only in the male. And paternal imprinting can also happen where certain genes are silenced in the male. We can only see the expression in the female. So if a gamete harboring the maternal imprinted gene combines with a gamete harboring the paternal imprinting gene for the same gene, then it may have a deletion equivalent to the gene silencing, so there will be no gene product. It can be. So the mother and the father have distinct set of imprinted genes depending on the DNA methylation patterns in the genes. And it happens during gametogenesis. The DNA methylation marks are transferred to the gametes. So genomic imprinting involves sex-specific methylation is true. Now let us look at Prader-Willi and Angelman syndrome. These are two diseases, and these diseases occur with a imprinting of same region on chromosome 15 has been implicated for both Angelman and Prader-Willi syndromes. So here pink represents maternal copy and blue is the paternal copy. In Prader-Willi syndrome, loss of maternal contribution that is linked to Angelman syndrome. Angelman syndrome, we have loss of maternal loss of maternal contribution, and the loss of paternal contribution that is linked to PWS. So paternal silencing, Prader-Willi, and maternal silencing leads to Angelman in the same chromosome. Prader-Willi syndrome is caused by deletion in the paternal copy of a gene, while Angelman syndrome is caused by deletion in the maternal copy. So there is a deletion which is not imprinted, so the deletion can be silenced. And now, since the silencing genomic imprinting has failed for the paternal copy, it will cause Prader-Willi syndrome, and if the imprinting has failed for the maternal copy, it can call Angelman syndrome. So the statement 3 is also correct. Prader-Willi syndrome caused by deletion in the paternal copy of the gene, while Angelman syndrome caused by deletion in the maternal copy of the gene. So option B is the correct answer. 1 and 3 only. So with this we come to the end of the problem set solving session corresponding to lectures of week 2, and I hope we could learn something or revise something from the lectures. We could learn something new and also practice the style of answering questions, because I'll stop sharing my screen now. Yeah, and as per the request, I'll quickly open my YouTube channel. So all these this session is also recorded, and the previous session of problem solving has also been recorded. So I'll just put up the YouTube link here in the chat box. You can readily go and watch that to clear your doubts. I have solved some similar questions, and just wait a minute. So I have created a playlist which will have all the so this is the playlist. I'll keep uploading the future problem solving sessions in this playlist. Here is the link for recordings of problem solving session. You can access this link here. You will also get this link via the NPTEL team. NPTEL team will provide you with this, as well as a Google Drive link which will contain these slides which I have prepared for you guys. So even if you have missed it, you can go back and look at it, have a have a thorough understanding, and please keep up with your lectures. It will always help you in the long run. Hope to see you in the next class where we'll be solving problem set 3 corresponding to the lecture of week 3. So be prepared and let's have a happy evening. Bye bye. I hope you are all doing well. I'll end this session now and you may leave the meeting. Thank you.